Sea urchin chemotaxis. One molecule, chemotactile molecule, a 14 amino acid peptide. 14 amino acid peptide called Resact. Resact has been isolated from the egg jelly of the sea urchin, Arbacia punctulate. Resact diffuses readily in seawater and has a profound effect on very low concentrations when added to suspension of Arbacia sperm. Resact is specific for A. punctulata and does not attract sperm of other species. A punctulata sperm have receptors on their plasma membrane and bind the Resact, which can swim up a concentration gradient of this compound until they reach the egg. Resact also acts as a sperm activating peptide. Sperm activating peptides cause dramatic and immediate increase in mitochondrial respiration and sperm motility. The sperm receptor for Resact is a transmembrane protein, and when it binds Resact on the extracellular side, a conformational change on the cytoplasmic side activates the receptor's enzymatic activity. This activates the mitochondrial ATP generating apparatus as well as the dynein ATPase that stimulates flagella movement in the sperm. Next, the acrosomal reaction in sea urchins. A second interaction between sperm and egg is the acrosomal reaction. In most marine invertebrates, the acrosomal reaction has two components: the fusion of the acrosomal vesicle with the sperm plasma membrane and exocytosis that results in the release of the contents of the acrosomal vesicle and the extension of the acrosomal process. The acrosomal reaction in sea urchins is initiated by contact of the sperm with allows calcium to enter into the sperm head. The exocytosis of acrosomal vesicle is caused by the calcium-mediated fusion of the acrosomal membrane with the adjacent sperm plasma membrane. The egg jelly factors that initiate the acrosomal reaction in sea urchins are often highly specific to each species. Then species-specific recognition in sea urchins. Once the sea urchin sperm has penetrated the egg jelly, the acrosomal process of the sperm contacts the surface of the egg. A major species-specific recognition step occurs at this point. The acrosomal protein mediating this recognition is called bindin. Acrosomal protein, remember, that is present on the acrosome. Then in 1977, Riquier and co-workers isolated this non-soluble 30,500 Dalton protein from the acrosome of Strongophoscentratus purpuratus and found it to be capable of binding to degellied eggs of the same species. Further, its interaction with the eggs is relatively species-specific. Bindin, isolated from the acrosomes of S. purpuratus, binds to its own degellied eggs but not to those of another species. In mammals, Zona pellucida is there, we know. The zona pellucida in mammals plays a role analogous to that of the vitelline envelope in invertebrates. This glycoprotein matrix, which is synthesized and secreted by the growing oocyte, plays two major roles during fertilization. It binds the sperm and initiates the acrosomal reaction after the sperm is bound. The binding of sperm to the zona is relatively but not absolutely species-specific. Species-specific gamete recognition is not a major problem when fertilization occurs internally. The binding of mouse sperm to the mouse zona pellucida can be inhibited by first incubating the sperm with zona glycoproteins. Then Blail and Wasserman in 1980 to 1986 to 1988 isolated an 83 kilodalton glycoprotein ZP3 from the mouse zona that was the active competitor for binding in this inhibition assay. The other two zona glycoproteins they found, ZP1 and ZP2, failed to compete for sperm binding. Moreover, they found that radio-labeled ZP3 bound to the heads of mouse sperm with intact acrosomes. Thus, ZP3 is the specific glycoprotein in mouse zona pellucida to which the sperm bind. ZP3 also initiates a acrosome reaction after sperm have bound to it. The mouse sperm can thereby concentrate its proteolytic enzymes directly at the point of attachment at the zona pellucida. The molecular mechanism by which the zona pellucida and the mammalian sperm recognize each other is presently being studied. The current hypothesis of mammalian gamete binding postulates a set of proteins on the sperm capable of recognizing specific carbohydrate regions of ZP3. Removal of this threonine or serine-linked carbohydrate groups from ZP3 abolishes its ability to bind sperm. So, several proteins have been identified on the sperm cell surface that specifically bind to the ZP3 carbohydrates. Moreover, the deletion of these proteins from the sperm can inhibit and eliminate sperm zona binding. Then, introduction of the mammalian acrosomal reaction by ZP3. Unlike the sea urchin acrosomal reaction, the acrosomal reaction in mammals occurs only after the sperm has bound to the zona pellucida. Mouse sperm acrosomal reaction is induced by the cross-linking of ZP3 with the receptors for it on the sperm. This cross-linking opens calcium channels to increase the concentration of calcium in the sperm. The mechanism by which ZP3 induces the opening of the calcium channels and the subsequent exocytosis of the acrosome remains controversial, but it may involve the receptors activating a cation channel for sodium, potassium, or calcium, which would change the resting potential of the sperm plasma membrane. The calcium channels in the membrane would be sensitive to this change in membrane potential, allowing calcium to enter the sperm. Then, secondary binding of sperm to the zona pellucida. During the acrosomal reaction, the anterior portion of the sperm plasma membrane is shed from the sperm. This region is where the ZP3 binding proteins are located, and yet the sperm must still remain bound to the zona in order to lace a path through it. In mice, it appears that secondary binding to the zona is accomplished by proteins in the inner acrosomal membrane that bind specifically to ZP2, whereas acrosome intact sperm will not bind to ZP2. Acrosome reacted sperm will. So, moreover, antibodies against the ZP2 glycoprotein will not prevent the binding of acrosome intact sperm to the zona, but will inhibit the attachment of acrosome reacted sperm. The structure of the zona consists of repeating units of ZP3 and ZP2, occasionally cross-linked by ZP1. It appears that the acrosome reacted sperm transfer their binding from ZP3 to the adjacent ZP2 molecules. After a mouse sperm has entered the egg cortical granule release their contents. One of the proteins released by these granules is a protease and specifically alters ZP2. This inhibits other acrosome reacted sperm from moving closer towards the egg. In guinea pig, secondary binding of the zona is thought to be mediated by the protein PH20. Moreover, when this inner acrosome membrane protein was injected into adult male or female guinea pigs, 100% of them become sterile for several months. The blood sera of these sterile guinea pigs had extremely high concentrations of antibodies to PH20. The antiserum from guinea pigs sterilized in this manner not only bound specifically to PH20 but also blocked sperm zona adhesion in vitro. The contraceptive effect lasted several months, after which fertility was restored. These experiments show that the principle of immunological contraception is well founded. Then, some other aspects related to the capacitation. The reproductive tract of female mammals plays a very active role in mammalian fertilization process. While sperm motility is required for mouse sperm to encounter the egg once it is in the oviduct, sperm motility is probably a minor factor in getting the sperm into the oviduct in the first place. Sperm are found in the oviduct of mice, hamsters, guinea pigs, cows, and humans within 30 minutes of sperm deposition in the vagina. Rather, the sperm appear to be transported to the oviduct by the muscular activity of the uterus. By whatever means, mammalian sperm pass through the uterus and oviduct, interacting with the cells and secretions of the female reproductive tract as they do so. These interactions are crucial for their ability to interact with the egg. Newly ejected mammalian sperm are unable to undergo the acrosomal reaction without residing for some time in the female reproductive tract. The set of physiological changes that allow the sperm to be competent to fertilize the egg is called capacitation. The requirement for capacitation varies from species to species. The molecular changes that account for capacitation are still unknown, but there are four sets of molecular changes that may be important. First, the fluidity of the sperm plasma membrane is altered by the removal of cholesterol by albumin proteins found in the female reproductive tract. Second, particular proteins or carbohydrates on the sperm surface are lost during capacitation. It is possible that these compounds block the recognition sites for the proteins that bind to the zona pellucida. Third, the membrane potential of the sperm belong more negative as potassium ions leave the sperm. These changes in membrane potential may allow calcium channels to be opened and permit calcium to enter the sperm. So, calcium and bicarbonate ions may be critical in activating CAMP production and in facilitating the membrane fusion events of the acrosomal reaction. Fourth, protein phosphorylation occurs. Moreover, it is still certain whether these events are independent of one another and to what extent each of them cause sperm capacitation. Then gamete fusion and prevention of polyspermy. Fusion of the egg and sperm plasma membranes. Recognition of sperm by the vitelline envelope of zona pellucida is followed by the lysis of that portion of the envelope of zona in the region of the sperm head by the acrosomal enzymes. This lysis is followed by the fusion of the sperm plasma membrane with the plasma membrane of the egg. Then, sperm egg binding appears to cause the extension of several microvilli to form the fertilization cone. Homology between the egg and the sperm is again demonstrated because the transitory fertilization cone, like the acrosomal process, appears to be extended by the polymerization of actin. The sperm and egg membrane proteins then join together and material from the sperm membrane can be found on egg membrane. The sperm nucleus and tail pass through the resulting cytoplasmic bridge, which is widened by the actin polymerization. A similar process occurs during the fusion of mammalian gametes. In sea urchin, all regions of the egg plasma membrane are capable of fusing with sperm. Fusion is an active process often mediated by specific fusogenic proteins. It seems that bindin plays a second role as a fusogenic protein.